And you can't lean forward. Are you okay with that?
Yeah. I can move down too.
Kate, are you going to set up Skype on, or are you still going to YouTube it, right?
It's live right now.
I'm just
Oh,
on my
okay.
phone right now. It's my computer is not plugged in, but I switched to Skyping you.
We have it live on this, but we can set up Jeff's computer with
I
Skype.
can switch to Skyping you on my computer after I get the introduction. Are you there?
I'm gonna.
That doesn't bode well.
Should I pause it or just live?
You could just, I mean, or you could put the little cover over
Then
it.
people will think it's not working.
Oh, that's true. Can it go next to your
Yeah.
You have like your face that you put on that.
Oh my god,
This is the most recorded PhD talk.
we
We
should
now
put a
have 3
pony-
separate
now have-
recordings.
Oh my god, we should put a ponytail on that.
Hashtag millennial PhD.
You guys are millennials, are you?
Yeah,
She definitely is, and I'm right on the cusp.
You are.
Noah is not.
It was
Apparently
19,
it's
it's
like-
1982.
Well, the way that they do it is, did you have a cell phone in high school?
It's actually by year. It starts in 1982. It's considered a millennial.
Okay, so that's it, whatever.
Do you have a cell phone?
I got my first cell phone in. So yeah.
I got mine as soon as I graduated.
I didn't have my first cell phone after my PhD.
There's also about here, they started deciding that there should be another name for kids that were 80s and early 90s. So you like remember when internet wasn't a thing and like. Yeah. Otherwise, it could be like.
Very excited.
Really excited.
All right. You see if Stuart picks up?
Did you already call Stuart? Should we all stand here and just stare? Oh yeah, the end step director wish me good luck.
Stuart Sandin is online.
Went out of the hotel, so the internet cut out.
Okay, you can get the internet in the hotel
Okay,
now.
yeah. Hi.
Yeah, I mean, we could prop it
Well,
if it
it's-
doesn't come back up in time. We can prop it.
It's live, so if he gets internet at any point,
You can click on the live sheet. Just start to close this one. 10 o'clock. Maybe get it like 2 minutes. Okay.
Okay.
Like Cole said, we can hear you loud and clear and do hi Jen, my friend hugs. Cole. I'm like, I know. There's John. We were like,
Guess it's time.
it's time.
Well, good morning, everyone. Thank you all for joining us this morning. It's my great pleasure to introduce Miss Amanda Carter for her PhD thesis defense. So, it's clear that from a young age, Amanda was exposed to the ocean quite frequently because her mom, Celeste, had also a passion for the ocean and for the sciences. Amanda grew up in Los Gatos, where she spent a lot of time in the ocean around Santa Cruz and frequently spent the summers in Maine, which we all know has a beautiful ocean. So she may have had an early stint as a sailor, thinking about this as a possible career path, but the career in sailing gave way rapidly to a career in lifeguarding. But unfortunately, Amanda liked the rubber ducky a little bit too much, so that didn't work out either. Either way, as I mentioned, Amanda spent a lot of time with her mom in and around the ocean, growing up around Santa Cruz, exploring the coasts and the intertidal, as well as in Maine. And she took a brief stint where she thought she was going to be a ballerina and then maybe a professional equestrian and then a vet. But it's clear from this last photo that she was had an early destiny to be in my lab. She's adorned with a seaweed necklace, and so that just set up her path for the future, where she joined my team, where we like to adorn ourselves with seaweed all the time. So Amanda came down to San Diego for her bachelor's degree. She joined my lab pretty much right away during her bachelor's degree as a lab volunteer. But when she came down to San Diego, she actually kind of followed some family history and tradition. In fact, her family has a long history. They were actually lighthouse keepers here in San Diego back in, I don't even know what year that was. And apparently Amanda's great uncle was a close friend of Carl's Hubs, where they spent a lot of time diving and surfing together in San Diego. So interestingly, Amanda followed her family back to San Diego to follow the passion of studying marine science. So Amanda, as I said, got her bachelor's at UC San Diego. She also got her master's at UC San Diego, and then she became a lab manager at UC San Diego, and now is doing her PhD at UC San Diego. So I like to call this Amanda's tenure, or her 10-year stint in the SMID lab at UC San Diego. Along the way, though, she, through her PhD, she received an EPA STAR Fellowship, which is a prestigious fellowship to conduct her PhD research. And over these 10 years, Amanda has had many wonderful experiences. She joined a dynamic group of lab members, both in my lab and Stuart Salmon's lab. She traveled the world, participating in conferences, conducting research, making lots of friends, colleagues, and collaborators around the world. And one thing that you'll notice in all of these photos is that Amanda's always smiling. I could not find an embarrassing picture of Amanda. She's always happy and always smiling. And I think this is an important testament to her personality. Amanda's always a really positive person. She is always a glass half full kind of person. She's always looking at the bright side, cheering up her friends, and just being a generally positive member of the lab who, again, is always smiling. Whether she be crunching data, drinking wine, or hanging out with friends, there is always a smile. However, I did find a couple pictures where I thought, and I thought this was funny, but then it became even more funny when I found that this face actually seems to be reoccurring. So she at least carries another face through her life history from her childhood to present day. Okay, all that being said, Amanda spent her time doing her PhD in my lab with the very difficult path of having to take a private jet down to this wonderful remote pristine island in the middle of the Central Pacific, where she studied some of the coral reefs on one of our most remote and highly protected islands in the world. That's Palmyra. Amanda spent many years diving the beautiful reefs of Palmyra, but this ended up not being the primary focus of her thesis. In fact, she decided to study the most ugly reefs of Palmyra, and that's because these reefs are significantly impacted by this invasive species that needed a lot of research to try to understand the dynamics and what was going on. So Amanda became a corral morph warrior, where she spent countless hours underwater trying to kill this invasive species, but also trying to understand the ecological and kind of physiological, chemical interactions that the species has with associated reef organisms. Along the way, she became adept at conducting lab experiments and doing physiological measurements. She became a great boat driver, a great team member, a mentor, and an all-around very supportive and wonderful person to have in the lab. Even when things were rough, and those of you who don't know what's happening in this picture won't totally understand, but if you look at Mike's face, that just explains what's happening here. These guys spent nearly 3 weeks with this device getting towed around the reefs of Palmyra in very adverse conditions, and they were not very happy about it. But Amanda still kind of has a smile. She was always there for me as a student, willing to hold the umbrella for me to keep me out of the rain, even though we were soaking wet already. Being a great dive buddy, and even taking the responsibility of taking images for me for one of my long-term time series when I wasn't able to be on Palmyra, and just an all-around good friend and student. Amanda also got to travel, as I mentioned, to present some of her research. nationally and internationally, always smiling again along the way. And we know none of this would be possible without Amanda's furry friends, who we know all of us know she couldn't live without. Along her time doing her PhD, Amanda also met some very important people, including Jeff, who decided to propose to Amanda underwater. Very apropos, and they recently tied the knot. So also very wonderful experience to have while she was here at Scripps. Amanda met a lot of great friends, long-lasting relationships, and it seems that Amanda, Sam, and Lindsay really like to take pictures together. I found several, these are just a few. And as in normal kind of conditions in my lab, nothing would be the same without everybody dressing in ridiculous outfits every once in a while, and Amanda certainly embraced this, as you can see, and many different outfits. She really likes to dress up as a shark. Okay, so all joking aside, through Amanda's time here at Scripps doing her PhD, she currently has 5 peer-reviewed publications, a few others that are currently in review. She received numerous grants and awards for her PhD, including, as I mentioned, the EPA STAR Fellowship. She also received the Pfizer Family Foundation Fellowship. We received multiple grants to conduct the research that Amanda will present today. She's given many talks at conferences, and she's had extensive teaching and mentoring experience, kind of all setting her up for the next stage in life. And that is, Amanda was recently awarded or announced as one of the 2019 John A. Krause Marine Policy Fellows, which she will be starting in DC in February. So given all of that, and that time is ticking, I now would like to invite Amanda to come up and defend her PhD. Thank you.
All right, let's see if everything continues to work. Sweet. Okay, thank you, Jen, so much for that. I'm gonna try not to get emotional. I do want to say though, thank you all for coming here today. I've been at Scripps and at UC San Diego for a long time, as you heard, and it's just really awesome to see everyone here today. I've made a lot of friends along the way, and I'm just really grateful that you're all here. So without further ado, I'm here to talk to you guys today about the research I've been doing for the last few years, looking at the impacts of local and global stressors on benthic communities at Palmyra Atoll, as Jen was saying, and investigating the response to invasion and bleaching at multiple ecological scales. So I think that we've all heard a lot of coral reef talks probably during our time at Scripps. Even if you aren't a scientist here, you've probably heard about how important coral reefs are, how they're the tropical rainforests of the ocean, how much we rely on them for their biodiversity and their economic value. And unless you've been living under a rock or a coral for the last few years, you probably saw some of these headlines. This was the obituary for the Great Barrier Reef that came out a few years ago and received a lot of press. And if you were paying attention the day after Thanksgiving, if you were asleep from eating a lot of turkey, you might have seen that the 4th National Climate Assessment was released, where they actually specifically referenced coral reefs and the fact that the changes in our climate and these anthropogenic stressors that we're creating are leading to a loss in ecosystem services. And so, although I think it's really important that we all understand the impact that we're having on coral reefs, I also think it's really important to hear about places that perhaps are not turning into some of these images that you've seen here. And to really understand the impacts that we're having on these reefs and to understand their abilities to recover from these impacts, you have to understand what the driving factor is on a coral reef, or what that limiting resource is. So this is like stepping way back and thinking about this. And that is that coral reef dynamics are driven by competition for space. And this is a pretty fierce competition that happens on reefs. These are just a few images that I've taken over the years. This is coral versus coral, coral reef sheeting over algae. This is that invasive species Jen showed you guys images of This is one of my favorites. This is actually two corals of the same species that are just kind of fighting out for space on a reef. And this competition and the winners or losers of this competition create what we see and what we think of as the reef when you see photos of one. And so in what we would call a healthy reef ecosystem, or what is a calcifier-dominated ecosystem, these benthic dynamics favor the corals and the calcifying organisms that help create that reef. However, the stressors that you've been hearing about a lot in the news can actually shift those competitive dynamics and create a climate or an area that favors some of these non-calcifying organisms. And I'll be talking about a few of these different stressors today. But briefly, you can think about these in sort of four different areas. You have natural stressors, and then you also have human-induced, and then you have global and local stressors. And so a natural global stressor could be something like a huge hurricane or storm event coming through, killing a lot of coral and opening up a lot of space that other organisms can then settle on. It could be something like a local natural stressor like predation. This is a crown of thorns starfish, something that some of you may have heard about that can come through and rapidly feed on coral. Or it could be something like disease coming through and removing a large amount of the coral community. We also have local human-induced, things like overfishing, nutrient pollution, sedimentation, altering those dynamics, perhaps creating conditions that favor fleshy algae rather than calcifying organisms. And then also human-induced global stressors. So this is a lot of what you've been hearing about recently in the news. We have warming oceans, we have bleaching events, and then also ocean acidification, which is slowly dropping the pH of the ocean a little bit and making it harder for calcifying organisms like corals to make their skeletons. And so some of those competitive interactions of the stressors can create these disturbances or perturbations on a reef that lead to this shift in the dominant benthic organism on the reef. And so you can have this shift from reef builders, all these calcifying organisms, to fleshy algae or macroalgae instead. And the ability to study these shifts and to understand the trajectory of reefs after perturbations that can cause this kind of community transition requires that you're able to study the benthic dynamics and the trajectory of that reef over a long period of time. And so sometimes these transitions will happen and there might be multiple stressors on that reef. Perhaps they've removed a lot of the herbivores, those fish that breaks down that algae like little lawnmowers. Maybe there is a agricultural field above that reef, and every time it rains, sediment runs down and nutrients run down, and you actually get a more long-term shift in that community following a disturbance event where macroalgae or fleshy algae dominate. However, you also have instances where a reef experiences a disturbance that creates a change in that dynamic, but then it's able to recover if it doesn't have all of those other stressors impacting it throughout time. And so this is an example of a reef on the Great Barrier Reef in 2006, and you can see this is a very coral-dominated community with lots of big plating acropora colonies. It suffered a disturbance in 2010 and became strongly dominated by that fleshy macroalgae. In 2012, though, you see that that algae has decreased in abundance, and you can see there's actually a lot of little juvenile corals growing in on this reef. And then by 2014, so 8 years later, this reef now looks a lot like what it looked like in 2006. And this is a really important trajectory to study and to understand. But if you think about this, if you went at any one of these 4 years and you didn't watch what changes on this reef at that time, you might think that, well, this is a really degraded reef. It obviously had a lot of stressors here. It's not really an area worth studying. And you wouldn't capture this full trajectory throughout time. So to understand these sort of trajectories and to study reefs where they might have this ability to recover after these disturbances, you have to go to places that don't have all those multiple confounding stressors that I was talking about before. And so to do that, as Jen said, this was very rough having to travel down to this island. I used Palmyra Atoll as a case study for my PhD. As Jen said, it's one of the most remote and uninhabited islands. It's about 1,000 miles south of Hawaii, and it's never had a local human population. So although there have been some impacts there, it was occupied during World War II, Palmyra is a relatively intact and unaffected or unimpacted ecosystem where we can study its response to different stressors. And Palmyra has a relatively small group of schools that go down there and conduct research. It has beautiful reefs. This is really my happy place. I think a lot of you guys that know me know this. They have healthy fish populations, lots of sharks. It's really beautiful. And perhaps most importantly is the fact that Jen has actually been going to Palmyra since 2005 and annually since 2009. And so you don't have to worry about all these names, but these are a lot of long-term monitoring sites at that atoll. So how I was saying, you need to have that monitoring throughout time to capture those changes in reef dynamics. We're able to capture that at Palmyra because of the long-term monitoring data set that we have thanks to Jen and Stuart and our labs. And just to give you an idea of what the reef looks like, it's just a gratuitous, pretty video. But it is really important to see this. This is an example of a site in Palmyra that is a heavily calcifier-dominated area. You can see that this is coral on top of coral, and some of those images that I showed of the coral battling coral are taken from this site. So it's an area where calcifiers are clearly winning. However, despite these really beautiful sites at Palmyra, it isn't completely free of some anthropogenic impacts. And in 1991, a shipwreck was deliberately grounded on the reef terrace at Palmyra Atoll. You can see it here, a big long-lining vessel. There was a bloom of cyanobacteria after this wreck occurred. And then this invader that you can see in the foreground here began expanding across the reef terrace. And this is an image from Terry Work's paper in 2008 showing the epicenter of that shipwreck and just the radiating tracks of that invader moving out from that site. And Jen actually showed a great photo from one of the sites at Palmyra where you were able to see this, but that previously calcifier-dominated reef turned into something like this. So it was completely covered, areas up to 100% cover of this invader. And I have avoided mentioning this name because this always brings me to the next question. So this invasive species is called a corallomorph. And whenever I talk to people about my work, it's inevitable that I get a, what's a corallomorph? And then normally people can't say corallomorph because it sounds too much like coral. And interestingly, corallomorphs are actually Nigerians. They're anthozoan, so they're related to coral. But they're a fleshy, Jen's gonna hate me for saying this, but Jen likes to think of them as fuzzy donuts, which is still one of my favorite descriptions for them. So they're this fleshy organism. They don't have a calcified structure. And this allows them to grow rapidly, and they can proliferate and do well in some disturbed environments, and it demonstrates this competitive advantage over corals. So this is a local stressor at Palmyra that we are able to capture and study in that long-term monitoring data set. And then, unfortunately or fortunately for my research, we were also able to capture the response of the community to the 2015 and 16 Global Coral Reef Bleaching event. This is an image of the temperature on the reef terrace, which is that shallower area of Palmyra, and the core reef, which I'll be talking about. Those are at about 5 and 10 meters depth, taken from in situ sensors that Jen's lab has had out on the reef for many years, and that we work on every year to get this change in the temperature at depth throughout time. And you can see this dashed line is the Palmyra-specific coral bleaching threshold. And you can see that in 2015, from about April to November, the temperatures at Palmyra were elevated above that bleaching threshold. And indeed, we saw widespread bleaching at Palmyra. However, we actually saw really low mortality, and that created a unique opportunity to look at a global stressor as well. So taking a step back, the general overview of what I'll be talking to you guys about today is here. So in chapter 1, we'll be thinking about what the spatial and temporal dynamics are of Rhodactis howesi populations at Palmyra Atoll. Then we'll think about what competitive mechanisms that invader might be using to outcompete corals at the atoll. And then we'll think about whether or not we see any species-specific responses in that coral community that might signify the resilience to that invader, or perhaps differing responses to overgrowth by the corallomorph. And then in chapter 4, we'll think about species-specific coral microbial community response to and from bleaching, or to and after bleaching. So for chapter 1, we'll start with the spatial and temporal dynamics. And I kind of like to set this in a framework of going from macro to micro throughout my PhD. So I started big, thinking about spatial ecology around the island. And then I got a little bit smaller and started thinking about the chemical ecology of the invasion and that competitive interaction between the coral and the crownwork. And then I got really, really small, and I went back to my mom's roots, because my mom's a microbiologist. And I thought about the changes in the microbial community associated with invasion and also with bleaching. But we'll start with chapter 1, that macro or large scale view. So this paper is changes in benthic community composition associated with the outbreak of the Crownworm housi at Palmyra Atoll. It's currently in review at Coral Reefs. Hopefully, just got our last round of reviews, and this will be published shortly. And just to give you a brief overview of what I'm going to be talking to you about in this chapter, we're going to think about where is it? So where have we observed this crownworm? How are the benthic communities and the crownworm populations changing around the atoll? How are they changing at the shipwreck site, or that epicenter of the invasion? And then we're going to think about whether or not certain substrate or benthic communities are more easily invaded. And to think about this, we use a really large scale and long-term data set that Jen's lab has been collecting for the last decade. And this includes toad diver surveys. This is actually not the toad board that Jen showed in that earlier image, but it's the same idea where you get towed along the reef and look for presence of the crownworm. We used photo mosaics, which are compilations of thousands of images taken by the Salmon Lab and stitched together to create these 100-meter squared swaths of the reef that you can actually zoom in on and look at one coral colony or a centimeter of that coral colony throughout time. And then we also use photo quadrats, which Jen mentioned. And so these are those 1 meter squared photos that are taken in the same place throughout time from year to year so that we can track some of those smaller scale changes in the benthic community. And we also were able to capture in this long-term data set the actual removal of that longliner wreck. So the U.S. Fish and Wildlife Service received money to take that wreck off of the reef terrace in the winter of 2013. And so in the fall of 2013, colleagues and I set up, here I am putting in a permanent photopod station. We put in 4 cardinal direction transects radiating out from that shipwreck site, or 3, one to complement one Jen had there before. And so throughout this talk, I'm going to be referencing some functional groups of these key benthic players on the reef. And just to orient you guys to them, we have the crownworm, shown here, that I talked about. We have our hard corals, so the reef-building corals that most people associate with the coral reef. We also have crustose coralline algae, or what I'll call CCA. And this is an important calcifying organism on the reef. It looks like bubblegum, and it helps cement coral rubble together and also provides some important settlement cues for juvenile corals. We also have mixed turf assemblages. So these are those small, short, little assemblages of algae that a lot of herbivores or fish like to graze down, and then the larger macroalgae as well. So coming back to that first objective, we wanted to know, where is the crownworm? Where have we seen it around Palmyra in this decade-long monitoring set? And you can see here, this is that epicenter of the shipwreck, and these are toad diver surveys that we conducted to complement the work done by 3 working colleagues in 2008. And you can see that the crownworm has followed flow patterns pretty well, moving along the southern coral reef at the atoll, but then we've also started seeing it at some of these northern sites as well. And I'll be talking about some of Jen's long-term sites. You don't have to worry about names, but this is just to give you an idea of where they are in reference to that shipwreck site. And so we wanted to think about how those benthic communities around the atoll are changing. We've seen that the crownworm has spread to additional sites away from that original epicenter of the shipwreck invasion. And so to do that, we used data from Jen's permanent photoquadrat monitoring transects from 2009 to 2017. And so this is showing you just the change in the crownworm at first. These are the 4 permanent monitoring sites that have crownworm present in those photoquadrats. And you can see that there's 2 really distinct things that we're seeing around the island. At 2 of the 4 sites, we've been seeing this increase in the crownworm. And indeed, at this site, it's reached over 40% cover, and it seems to be continuing to expand there. In contrast to that, though, we have 2 sites where we saw this initial bloom of the crownworm, and then it's actually been decreasing over time. And just to give you an idea of what that initial invasion that you can see captured in some of these sites look like, here's one of those photo quadrats, and that arrow is actually one single crownworm polyp that has landed in that photo quadrat. You can see lots of this pink CCA in here. And then now it's turned into 3 or 4 polyps. By 2014, it was around a third of this photo, and you can see that it's continuing to expand and grow across this photo. However, in contrast to that, we're seeing a really different story at the epicenter of that invasion. So here is that same, oh, actually, I should show you guys this really quick. Just to give you an idea, this is the final minus initial cover of all the benefit functional groups that I was talking to you guys about from sites around the atoll. And you can see that where we have that increase in the corral mark cover, that's been associated with a decrease in hard coral cover and also in turf at some of those sites. But we've seen a really different story at the longliner site, so where that shipwreck first occurred. Here, you can just take a look at the corral morph. So on average, we had around 70% cover of the corral morph in all of those photo quadrats from those 4 cardinal direction sites. And by 2017, we have less than 10% cover of the corral morph in those photo quadrats. So that's a 4-year time period, and that's a huge difference. And reassuringly, we've also seen this increase. This is CCA, that pink bubblegum pigment that I was talking about. We've watched that come back as the corral morph has decreased. And this is really important. I mentioned that it helps cement rubble together on the reef. And where the corral morph has had such high cover for such a long time, the reef is a little bit degraded, it's a little bit crumbly, and CCA has come in and really helped glue that back together, allowing for hard coral to actually start settling. And we have some really heartening settlement of hard coral recovering in that area and juveniles as well. And just to sort of visually show you what that decrease has looked like, here's one of those photo quadrats from 2013 throughout time. And you can see it was 100% corral morph cover in 2013. And now in 2016, you're actually hard-pressed to find a corral morph polyp in that image. So we see really different dynamics in this invasion around the atoll. And so this made us kind of wonder what drives the difference between these sites. So why do we see the corral morph take off at some sites? Why do we see it bloom at some sites and then actually stay static or decrease in cover? And then why do we see this drastic decrease in it at the longliner site itself? So to think about this, I looked at substrate preference for Rhodactis housei. And so this is that establishment that I mentioned before of that single polyp when it lands and it's able to survive, and then it can continue growing. And so I looked at every instance in all of our photo quadrat images throughout time where a single corral morph polyp landed. And then I also looked at what we would expect the corral morph polyp to land on based on the available substrate. So taking that cover of those photos and thinking about this is 50% coral and 50% this, how many times would we expect corral morph to land on one or the other? And we saw that corral morph actually strongly avoids settling on hard coral or is unable to survive to the point where it would be a juvenile polyp or a polyp, not a juvenile, on hard coral. And instead we see that it seems to prefer landing on CCA, that pink pavement, and also on turf algae, that really short assemblage of algae. In contrast to that though, I like to think of this initial establishment or that initial settlement as the corral morph kind of getting its foot in the door in a site or in a photo quadrat. And after it has that proverbial foot in the door, we see that it's able to overgrow almost all of those functional groups that we're talking about. So it may not land and survive on hard coral initially, but once it's started expanding that population, it is able to grow over hard coral, CCA, macroalgae, and turf algae. And so this has some important implications when you're thinking about the benthic communities at a lot of these sites around the atoll. You can think about areas that perhaps are more at risk of having juvenile corral morphs land and establish themselves there. And then you can think about sites that perhaps would have a bit of natural resistance to invasion by the corral morph. And so in summer, we saw that Rhodactis housei was present at many sites around the atoll, and we saw but in contrast to this, we saw a drastic decrease at the epicenter of invasion. This decrease was accompanied by the recovery of the benthic community at that site. We saw the increase in crustose coral and algae, that CCA, as well as a slight increase in hard coral. And we also found that Rhodactis housei preferentially settles in CCA and turf algae, avoids coral for that initial settlement, but then is able to overgrow any of those functional groups throughout time. So when you think back to that image I showed before, where there were some of those sites where corral morphs sort of bloomed, but then the population wasn't able to take hold, those are some of the really high coral cover sites of the island. And so I think that one of the really cool findings from this is the fact that it suggests that areas with really high coral cover have a little bit of natural resistance to this invasion, because the crown morph through sexual reproduction isn't going to have as much success as those little juveniles are looking for a place to land because they don't have as much substrate available to them. But sites where we see that bloom that kind of is increasing are actually areas where there's really high CCA and turf algae cover, suggesting that that's giving the crown morph an easy way to grow over that bedpost and to continue its invasion in those areas. So when we think about those areas where the crown morph is continuing to grow, sort of an obvious question is, how's the crown morph growing over hard coral? Right? We saw that it can grow over that functional group, but hard coral has a variety of different defense mechanisms and can be the competitive dominant on many reefs. So how's the crown morph doing this? And that brings us to chapter 2, where we're thinking about what are the competitive mechanisms involved in the spread of Rhodactis housei at Palmyra. And to orient you on our little macro to micro scale, we're now at that chemical ecology or the competitive mechanism between hard corals and the crown morph. So I started this talk telling you guys about competition on coral reefs, coral versus coral, who wins, who loses. And there's a variety of different competitive mechanisms that come into play when different benthic organisms are competing with one another. You have indirect competition, like taking up space that's available. I mentioned if a bleaching event or a crown of thorns or something comes through, you have a lot of space that used to be hard coral cover that's now open. And so you can actually compete with other benthic organisms indirectly by taking up that space before someone else can. We also have more direct forms of competition, like actually growing over your competitors, perhaps stinging them using nematocyst cells, or what I'll be talking about today, which is this chemical warfare, where secondary metabolites from these benthic organisms actually cause declines in the coral health or the other benthic organism's health and give a competitive advantage to the other organism. And so this has been studied quite a bit, thinking about allelopathic compounds from macroalgae and the role that they play in their competition with corals. And this is an image from an experiment done by Doug Rasher, where he extracted chemical compounds from macroalgae that you find on reefs, suspended them in these little strips and exposed coral to them. And he was able to show that without any physical contact between macroalgae and the coral, these chemicals extracted from the algae were able to cause tissue necrosis and ultimately lead to coral mortality. And if you think about that in terms of an invasion, you can see how some of those competitive mechanisms might assist in the expansion. So if you have a large area of space open up on a reef and you're able to settle quickly, like a weedy species, and then in addition to that, you produce some sort of compound that is not good for corals, and that's going to give you this competitive advantage, you might be able to grow quickly in these disturbed environments and rapidly take over. So we wanted to see whether or not there was any evidence of that chemical warfare happening between coral morphs and corals. And so chapter 2 was looking for evidence of allelopathy and competitive interactions between Rhodactis housei and Scleractinian corals. And specifically, we wanted to see if there was any evidence of that allelopathic interaction without the physical presence of the crown morph. And no one has looked at this before. People have looked at the competitive advantages of the crown morph, but they've never studied what that actual mechanism is. And if so, what part of that chemical fraction or chemical extract from the crown morph exhibits the highest bioactivity? And the end result of that would be to potentially identify what compounds are present in the chemical extract and what compound might be responsible for causing that tissue death. So as I mentioned, people have looked at some of the competitive abilities of Rhodactis housei. Primarily, they've thought of this in disturbed or stressful environments where the crown morph is able to rapidly take up space. It's a very G species. They can reproduce both sexually and asexually, so they're able to take up that space quickly. And in addition to this, Terry work in that 2008 paper at Palmyra also showed that exposure to the stinging nematocyst cell from the crown morph resulted in coral tissue death. And you can see this interaction between crown morph and corals as they grow over them. So this got me thinking about what what's causing this? Is this just the physical contact of the coral getting stung that's causing this tissue to die off, or is there some sort of chemical interaction happening here? So to test this, we developed this chemical bioassay following some procedures that were used by Doug Rasher. I showed his macroalgae experiment earlier, where we made extracts from the dried coralmorph tissue, which smells terrible, by the way. And then Chambers will never forgive me for making his lab smell like that. We suspended those extracts in phytogel strips, so these are like little agar strips. And then thank you to all the people that have helped me zip tie these very tiny and jelly and slippery strips onto very tiny pieces of coral because it's a surprisingly time-intensive process. But so we put these strips with the chemical extract in them, and we also have procedural controls that just have the solvent and no chemical in them. And we left them on my coral fragments for 72 hours. And we measured the coral health, or the, we don't want to use the word, or we frequently try to avoid using the word health, but here we're really thinking of it as the coral health, or what effect this chemical strip had on the coral's well-being. And to do this, we took images of those coral fragments before and after the experiment to look at the loss in tissue. And then we also used Jen's best friend, the diving cam, which is a fluorometer that measures the transport of electrons from photosystem 2 to photosystem 1 after a dark adaptation. And essentially what that means is that we looked at the photosynthetic efficiency or ability of the coral. And so if the coral is stressed by exposure to that coralmorph extract at the site under the strip, we're going to see a decrease in the maximum quantum yield for the photosynthetic efficiency of the coral. And thank goodness, we saw that. So here is an image of one of those treatment corals, and this is after removal of that strip. And it's pretty obvious and startling. You can see the amount of tissue loss that occurred under that strip, and that skeleton is actually exposed and that coral is completely bleached. In contrast, just so you can visually see this procedural control, the coral definitely wasn't happy about having a jelly strip that tied onto it for 72 hours, but the tissue loss observed was a lot less than that for the treatment. And indeed, we saw a significant decrease in that maximum quantum yield, or the photosynthetic efficiency of those treated corals, as well as a significant loss in their overall tissue cover. And so this gave us some of the first evidence that there was some sort of chemical extract or chemical present in the coralmorph that caused tissue necrosis without the physical presence of the coralmorph there. And so we further split that extract up into different compounds or into different fractions, and we repeated that experiment more times than are shown here. And we saw that the ethyl acetate fraction was more active than the methanol, meaning that the non-polar compounds in there, the guys that don't like water, were more active. And then we saw that after splitting that ethyl acetate extract up into these 5 different fractions, that the 25% ethyl acetate fraction was more active in this experiment. And then again, here we saw that the 100% ethyl acetate fraction was more active in this experiment. And so we took these two and with the assistance of Gabo and Chambers, looked at what was actually present in some of the highest abundance in those really bioactive fractions. And what we got was kind of weird. We found peridinin, which is a carotenoid, and which happens to be one of the most common carotenoids in dinoflagellates that are found in corals and in coralmorphs. And so we were kind of like, well, that's a bit weird. It makes sense that it might be in this extract, but it seems a bit odd that that might be something that's playing a role in this decline in the coral health that we're seeing. But interestingly, people have actually taken those peridinin-related carotenoids and they've used them in anti-cancer studies. They've shown strong antimicrobial activity. And they've also been shown to act as a virulence factor or a way for pathogens to have a vector into whatever the system may be, perhaps in this case, a coral. And so although we aren't 100% sure at this point that this is the chemical responsible for the tissue death that we're seeing, it did bring up some interesting questions and also made us think a little bit, if this has antimicrobial compounds or capabilities associated with it, what role might that be having in this interaction between the coralmorph and the coral? So in summary of this chapter, we saw clear evidence of a chemical interaction between the coralmorph and the hard coral,
We also saw that the two nonpolar fractions demonstrated the highest levels of bioactivity here. And we found that the most abundant compound present in those extracts was paradennin, that carotenoid that has been shown to demonstrate antimicrobial abilities. So now that we have found that compound and have seen that there is some sort of chemical interaction occurring between hard corals and the corallimorph, we decided to see whether or not there are any species or coral species specific responses in their microbial community to this invasion. And that brings us here to chapter 3, where we talk about the microbial ecology and microbial interaction occurring as corallimorph overgrows hard coral. And this is chapter 3, which is coral microbial community response to invasion by the corallimorph reductus haussai. And now I'm going to wow you with my artwork. This is a coral. And we frequently think about corals like this, right? It's this perhaps more visually appealing when you see it in person, but it's this skeleton that you see on the reef. It's beautiful, it creates this habitat. But the coral is actually not just this rock covered in animal. It also includes dinoflagellates, those symbiotic algae that live inside the coral tissue that are able to take energy from the sun and convert that into food for the coral. And there's also this very strong coral-associated bacterial community that live both on and in the coral tissues. And altogether, this is termed the coral holobiont. So I like to think about the coral holobiont as being sort of the molecular fingerprint of the coral. And the combination of the microbes, the symbionts, and the coral animal itself all give you this little fingerprint that you can take of a coral, and you can think about its status or how well it's doing. And you can also use this fingerprint to look at how the coral's molecular identity changes when it's stressed. And this is more of my beautiful artwork here. Some of you can actually probably identify these as species. And so we've seen that physical stressors, such as increasing temperature, decreasing pH, perhaps input of nutrients into the water, can lead to an increase in pathogenic bacteria in those coral microbial communities. And those pathogenic bacteria can actually replace the good bacteria that normally live on the coral surface. And those can cause changes in the coral's health. And a study done by my advisor in 2006 found that the exudates from algae can actually have this indirect effect on the coral microbial community that leads to coral mortality in the long run. And so corallimorphs are also very productive organisms. They photosynthesize, they're an animal as well. And so we were curious, as this corallimorph overgrows a coral colony, do they have an impact on the coral's microbial community throughout time? And as you move across this interaction zone from this unexposed tissue to the invaded tissue, do we see changes in those coral-associated bacteria? And could this have any implication for the coral health or the corallimorph's ability to overgrow these corals? So we looked at those shifts in the coral microbial communities during invasion by doing a large-scale in situ sampling effort in 2015 that was then repeated in 2016. This was at Homeyer as well. We took these samples at the shipwreck site and also at this back reef site, and these are the 5 different species of coral that we sampled. And I collected this along this little interaction zone here, 6 replicates moving from unexposed tissue to invaded tissue. And we did this for these 5 species. We took 8 colonies of each species, 6 replicates across each one of those colonies. We tagged that colony in 2015. I went back in 2016, refound that colony, and then resampled that interaction zone where the invasion is occurring. And this was a lot of time with me underwater with bone cutters and a tiny hammer and chisel, taking samples. And each one of those samples was placed into an individual WorldPack baggie. Here's Meg Johnson in my lab, where a past lab member wrangling about 50 baggies for me underwater as I'm putting samples into them. And so this was a massive effort, and it's always really nice when your massive efforts end up working. And so we saw that those coral microbial communities are significantly different from one another across species on that unexposed tissue, both in 2015 at that initial time point in the invasion, and also in 2016 as the invasion has progressed. This is an ordination plot here, and it's used to look at the similarity or dissimilarity between communities And here, for this sake, we're using this to look at the microbial communities of these 5 coral species shown here. And previous studies have shown that corals actually have very species-specific microbial communities. That coral holobiont can actually stay the same for a species throughout space and throughout time. And we did see evidence of that here at Palmyra on that unexposed coral tissue. But interestingly, as you move into that interaction zone, you see that the spread between these starts getting a little bit wider, and those microbial communities are looking a little bit more similar, although they were still significantly different from one another. But by the time you're in that invaded coral tissue, from those samples that I was showing you, these microbial communities across all species are no longer significantly different from one another. And this means that as the coralline morph overgrows that coral tissue, it actually causes a shift in the microbial community so that all of those microbial communities look similar to one another and have this sort of stressed molecular fingerprint. And interestingly, we saw that the similarity between all of those invaded tissues was largely driven by the presence of this, what's called others here, and this was this unclassified bacteria that we think is probably associated with the coralline morph itself. The similarity in the invaded tissues and then also in the interaction zones was also driven by a few families of bacteria that are frequently associated with stressed or unhealthy coral tissue. And then when you're thinking about specific species, we also saw that Montivora capitata, if you remember, I said in 2015 I tagged 8 colonies of coral. When I came back in 2016, half of those colonies of this species of coral were completely overgrown by coralline morph. I actually had to scrape coralline morph polyps off of those tags to find these coral colonies because I knew I wasn't crazy and that I knew where they were. And then I realized that they had been completely overgrown. And you can see that this sort of turquoise or mid-green bar is present on the healthy and that interaction zone tissue more as well, suggesting that perhaps this species is a bit more susceptible to overgrowth by the coralline morph. So in summary for this chapter, we saw that those unexposed coral communities were species-specific and significantly different from one another. However, invasion by the coralline morph led to this similar and stressed community throughout time. And that similarity between those stressed communities or those invaded communities was strongly driven by an unidentified bacteria that may be associated with the coralline morph and also by bacterial families that are commonly found on stressed out and unhealthy coral tissue. And so, as I mentioned before, we spent this time looking at this local stressor at Palmyra, but we also happened to be there during the bleaching event in 2015. And this gave us this really cool opportunity to also look at a global stressor on Palmyra, this reef that has minimal confounding impacts, and to look at the response of the coral communities there. And so I approached this using that same molecular fingerprint idea and comparing healthy coral colonies to bleached coral colonies in 2015, and then looking at how they recover in the following year. And so this brings us to chapter 4, which is how do the species-specific coral microbial communities respond to and recover from this bleaching event. And I feel like I should just mention here this beautiful artwork by Dr. Kate Kirby, showing the coral bleaching, and this chapter is titled Coral Microbial Community Response to a Bleaching Event at Palmyra Atoll. And so the 2 main objectives that I'll talk to you guys about today were to assess how did bleaching in 2015 shift those species-specific coral microbial communities, and then did that bleaching status in 2015, whether it was healthy or bleached, impact the recovery of the coral microbial community in 2016. So essentially, if a coral community bleached, did it recover back to what a healthy coral microbial community looked like, or was there some sort of change that was driven by that bleaching? And so we sampled 5 species of coral again. This time we sampled 4 healthy colonies in 2015 and 4 bleached colonies in 2015. Each one of those was tagged. We took 6 replicates across the coral tissue, and then we went back in 2016 and repeated this. And it's important to note that despite how widespread bleaching was on Palmyra in 2015, I actually did not lose a single one of the colonies that I tagged. There was some partial mortality, but there was no full mortality of all of these corals. And so here's that ordination plot again, looking at this. So just to have you guys orient yourself to the colors and shaped here. The turquoise color is the 2015 time point. The blue color is the 2016 time point. And then triangles are unbleached. And then the circles are bleached, or in 2016, those are the guys that were bleached in 2015 and then recovered and were resampled. So I'm breaking this down here by genus, actually, which I'll talk to you guys about a little bit more. These are our two acroporas. And you can see that the coral communities, the coral microbial communities in 2015 were significantly different from one another. So the healthy corals had one microbial community and the bleached corals had another microbial community. But interestingly, for this genus, we actually saw a recovery of those coral microbial communities in 2016. So even if they had bleached the previous year, we saw them recover back to that healthy or unbleached state the following year. And this contrasted pretty strongly with what we saw for the Montipora genus, these two guys here, where we saw this really strong clustering both by year and then also by health status. And so in 2016, if a coral microbial community had been bleached the year before, we actually saw that although the coral itself recovered and didn't die, there was this shift in that microbial community present. And we don't really know what the long-term implications are of that change in the coral microbial community, but it does suggest that this genus, or these two species sampled here, were a bit more susceptible to long-term changes as a result of that bleaching event. And then Postlepora meandrina, which people commonly think of as cauliflower coral, was kind of the in-between of these two. So we saw a significant difference between the bleached and healthy colonies in 2015, but they then returned to different states in 2016, but you can see that they didn't shift too much. So these circles were the bleached and recovered, and these triangles were the unbleached in both years. And these guys were also really interesting. They were the species that had some of the higher levels of partial mortality, but this is what that partial mortality looked like. So in 2015, this entire coral skeleton was alive. And when I came back in 2016, the cap of it was still alive, but we saw this partial mortality underneath. And so to sort of think about what those implications are, we saw that those species-specific responses actually fell along a pretty strong genus level. And this is really cool, and it might mean that perhaps there's some sort of innate taxonomic lineage in those microbial communities' response to thermal stress. And perhaps, although we don't necessarily know the implications of those colonies that changed their microbial community as a result of bleaching, that may have some sort of impact on their health long term. Perhaps those microbial communities have recovered to a more thermally tolerant one, or perhaps it's actually a negative recovery and those guys might have a decline in their coral health or difficulty responding to another bleaching event in the future. However, another thing that was really interesting here that I did mention before is that all of these coral colonies survived, as I said, and they also all maintain really high levels of those beneficial bacteria that are commonly associated with healthy corals. And when I got these results back, I spent a while, it's a lot of data, staring at it, wondering exactly how to tell the story, and I was describing it to my husband, Jeff, and I was like, yeah, well, we didn't see any of these big blooms in those pathogenic bacteria that are commonly associated with stressed or bleached corals. And I was like, I don't really know how I should tell this story. And he was like, well, it sounds like your story is what you didn't see rather than what you did see. And I was like, oh, you're brilliant. And so I really think that that's one of the biggest takeaways from this. We didn't see any full mortality of any of these coral colonies. And although they all experienced shifts in their bacterial community as a result of the stress of bleaching, we didn't see any blooms of these pathogenic bacteria coming into those coral microbial communities. And that may have played a role in why we saw the recovery of all of these coral colonies throughout time. So, just sort of a large scale wrap up for you guys. Palmyra was a really interesting place to study during my PhD. We saw the potential for recovery at Palmyra, both in response to a local disturbance of the crown morph and also to the global disturbance of that bleaching event. We saw low mortality, and we also saw that the community at that shipwreck site, or the epicenter of that invasion, was able to recover after the shipwreck was removed, and that community has actually returned to the calcifier-dominated state that it was before, even if it's not at a really high coral cover yet. And I think it's always a good thing when you finish your PhD and you find out that Sylvia Earle agrees with you. There was recently press that Dr. Sylvia Earle named Palmyra as a hotspot for the world's oceans because of the lack of these confounding stressors there and the ability of the reef to recover. And I actually had the pleasure of spending a week on Palmyra with Sylvia in 2016 when she was out there, and like life goals, she's 85 or 86 and was out diving on the forereef every day in inclement weather. And I had a really great time talking to her. She told me my nickname should be Amanda, which it already was, but I appreciated having Sylvia Earle reaffirm my nickname for me. So with that, I'd just like to thank so many people. I've been in this lab for such a long time. I've done a lot of work while here, and there are so many different people to thank. First and foremost, my committee. Jen, I would not be here or be the scientist that I am today without having been in your lab for the last decade. I really grew up in this lab. I started in here as an undergrad, and I've just gone through massive life shifts, and I really feel like Jen has kind of been my science mom and watched me grow up and had such a huge impact on forming me into the scientist that I am today. There is no one that I would rather get to dive on Palmyra with. She and I have so much fun underwater. Okay, and I'm not gonna start crying. Chambers, thank you for everything. I couldn't have done all this chemical ecology work and introduced me to the world of natural products. And to Gabo as well in his lab, who spent more time playing with ground work than I'm sure he ever wanted to. To Stuart, who hopefully is watching the live stream, excellent, in Jamaica, for always being willing to talk to me about statistics and dealing with me popping in his office at random times to ask him questions. Eric, you have been one of the most cheerful and wonderful committee members. Thank you. Always have a smile on your face. I always love getting to ask you questions about microbes that I know nothing about. And also to John, I actually TA'd for John 9 years ago. It was a long time ago. I TA'd for John for his ecology class in 2008 or 2009. This is a long time ago. Okay. It was probably last year. And I had a great time getting to continue working with him. Jen mentioned all of my funding sources before, but just an additional thank you. The EPA STAR Fellowship, which unfortunately is no longer around, was a really great funding resource for me as a PhD student. The Beister Family Foundation, the Center for Microbial Innovation up on Upper Campus provided me with all of the money to process the 2,000 samples that I brought back, and I would not have been able to do that on my own. It was a very expensive undertaking, and they also paid for part of my PhD, and to the U.S. Fish and Wildlife Service for the money that they've given Jen and our lab to do our work out at Palmyra. Also a huge shout out to the grad office, particularly Maureen, wherever you are. Thanks for coming to your first PhD defense. I just like, Maureen is the best. She handled my EPA STAR and a lot of my fellowships, and I've gone into her office and cried when I've been going through life events, and you guys are just the most supportive people for all of us here at Scripps, and we really appreciate you guys. To the Smith and Sandon lab family, I started to try and list people out, and it would have been like 5 slides. Thank you to all of you, past and present, for everything you've done, for all of my field buddies that have helped me out, we're so lucky to have the lab group that we have. It really is a big family, sometimes for better, sometimes for worse. But we all love each other, and at the end of the day, can all be there to help each other whenever we run into science problems or we just need to vent to one of our colleagues about something that's going wrong. Huge thanks to Luke Thompson, who's my co-author on my 2 microbial papers. This work couldn't have happened without him. He's been a fantastic co-author and collaborator to the CMI and to the night lab and to the people that help process some of those samples. Special shout out to the Palmyra Field Station staff. Spent the last 7 years of my life working out there, and the science on that atoll would not happen without them. And my brown feeds us all donut sharks that he makes by hand every morning, and it's amazing. And also a little special shout out to Peter for the thousands of LORs you've written me over the years. I am so glad I took your bio class as a master's student, which was like 9 years ago or 8 years ago, and you've just been a fantastic mentor and friend during my PhD. And also to Ron, oops, his name got cut off. But Ron, I took Ron's marine biology class as a sophomore at UCSD, and he's never been able to get rid of me. So he played a big part in my master's and point of being Hubs Hall's tenured student. And with that, just a special shout out to the Scripps community in general, to all of my friends that have been here throughout my master's, my PhD, undergrad. One of the absolute best things about going to school here is the community. I think it's just an amazing place to go to school. You make lasting friendships. You have amazing friends. TG is one of the best things ever. Like, no one gets that in grad school. And just thank you to all of you guys, to all of my field buddies I have over the years. I think I have more cute photos with Kate than I do with Jeff. Which also brings me to just a special thank you to particularly Kate and Sam and Lindsay, who isn't here. She's currently diving in the Caribbean. Lindsay was one of my first friends during undergrad. We've been friends for over 10 years and have stayed on a pretty similar path. And three of you guys are just some of the best friends I could ask for. And I love you guys. And of course, thanks to my husband, Jeff, for always being there. I'm really glad Christian and Rich, who I think are both here, paired us together for like 15 of our 18 dives during our scientific diver class. They talk a lot about how they mix things up, but they did not our year. And Jeff has just become my dive buddy for life. And I love you and our fur babies. And thank you for keeping me sane and feeding me and making sure I drink water throughout this whole process, and for being the best skipper I could ask for as well. And last but not least, thank you to my mom. You guys already saw some of these photos, including some other really embarrassing ones. But for those of you that don't know, my mom was also a scientist. She was also a single mom, and it was just her and I my whole life. And I grew up in her lab, making little dry ice bombs that I put underneath for students' chairs. I schooled DNA for my 5th grade science project and was disqualified from the science fair because it was too advanced. I'm so salty about that. And I literally would not be the person that I am today without my mom. You've been the most amazing role model and influence and mom I could ever ask for. So with that, I just want to everyone always ends with a sunset, and I figured I have 10 years worth of pretty epic sunsets, so might as well share some of them. And I'll take any questions. Hi, Peter.
Hi, Linda. Great talk. That was really fun.
Thank you.
In your last chapter, when you were talking about the interaction zone and the microbial community, the axes on your plot, I'm guessing were like principal component kinds of things, just related to the communities.
Exactly, yeah. So they don't necessarily have a
Right.
meaning, yeah.
And the first one explains the most variance, and the second one explains the
Yes.
second most. And I noticed that it looked like the bleached and unbleached seemed to separate more along the second axis than the first.
Yeah.
Which I'm guessing says that the species that are different between bleached and unbleached are relatively minor species.
I think that it varied a lot based on the different coral species in general. So not just the microbial species, but also the coral species. A lot of those differences were driven by different families for a lot of the different corals. That data set had over 450 different bacterial families present across all of them. And so there was a lot of variability in there. And I'd have to go in and look at the exact families for each one of those and look at what those OTU counts were for them to answer that or 100%. But the bleached and unbleached corals across most of the species, a lot of that difference was driven by presence of Rhodobacteriaceae and Enterobacteriaceae, which are both associated with bleached or unhealthy or stressed tissues, and the enterobacteria are actually strongly associated. They're copiotrophic, and so they're also seen a lot in areas where you have high levels of exudates in the water.
And I just had one other question. What was the ship made of that landed there?
Yeah, so it's funny that you asked that. That ship was something I did a lot of work on during my master's, and it was a I guess steel long lining vessel, but it is really interesting. I did some chemical or toxicity work looking at the shipwreck metal during my master's, and I found evidence of tributyl tin on the outside of it because it's an old vessel. It was from Taiwan. It was a long lining vessel. And so one of the things during my master's that I actually looked at was thinking about how metals leaching from that shipwreck might be facilitating the growth of the coralmorph or possibly inhibiting growth of the corals. And it was really interesting finding that tributyl tin was on there because that definitely could have been leaching off of the wreck and influencing that community. And also, Jen will remember this perhaps a little bit better than I, but I think the fuel tanks were still full or partially full with diesel when it was taken off of the reef as well, despite the fact that it had been there, it grounded in 91 and was removed in 2013.
I guess I was wondering about iron, but what's other things have
Yeah.
next year?
So iron leaching from the shipwreck was a hypothesis that was put forward and was a big part of the impetus for removing the wreck from the reef, the idea that it was this limiting nutrient that was facilitating the growth of the coralmorph and allowing it to outcompete corals. I focused on that a lot for my master's and looked at the metal content of coral and coralmorph at multiple sites around the atoll. And although I mentioned Palmyra has been uninhabited, it was occupied by the military during World War II, and there is metal everywhere. And so I actually found high levels of iron in both coral and coralmorph all around the atoll, and there wasn't a significant elevation in iron content of the coralmorph or corals at the longliner site. So it may have been playing a role in it, but I don't think that that was the primary driver. Like it wasn't like iron leaching out of that wreck, it was just letting the coralmorph go gangbusters and outcompete all the coral, just because there is a lot of iron present around Palmyra in general.
And is the thought that the coralmorphs were introduced with the ship, that they were attached to it or something? Or?
This is just like all these things that I thought about a lot for my master's. Yeah, it is a native species to the Indo-Pacific. Rhodocus haussai is native to the Indo-Pacific, but it demonstrates these invasive capabilities in disturbed environments. And so whether the coralmorph was introduced on the shipwreck or whether or not the shipwreck just came in and tore apart a bunch of the coral and opened up all of this space, and the coralmorph was just able to take off and occupy all of that available space. We're not 100% sure. I worked on the genetics of the coralmorph there with Ron, and we found that there was low variability in the population around Palmyra, but we didn't have another population to compare it to. So it was hard to say whether or not they were all coming from the point source of that shipwreck. And there are other sites around the island that have coralmorph that they look different than the haussai that you can see here. They're smaller, they're just little patches of it. They don't really change throughout time. And they don't seem to overgrow corals the way haussai is able to. Travis?
I had a kind of a question similar to Peter's. You showed a lot of competitive advantages of this coralmorph,
Yeah.
But then the coralmorphs drastically go away once the ship leaves.
Yeah.
So can you speculate as to why that might be happening if they have all these competitive advantages? They just go away.
Definitely. So the coralmorph went away at the shipwreck site, but it is increasing at those other sites, right? So but we did see this very different dynamic in the longliner site or that shipwreck site after that wreck was removed. And I think that could have a lot to do with the disturbance associated with the removal of that wreck. I showed those images a little bit of the operation, but that was a massive commercial marine operation. They brought in a huge barge, they put down massive pilings, they were cutting up the shipwreck underwater. It was a 110-foot long vessel, it wasn't small. And that created this big sedimentation event as they removed it. And I think that some of that initial decrease in the groundwork there might have been caused by the new pulse disturbance event of the removal of the shipwreck. And then that kind of just shifted the dynamics of that site so that the groundwork started declining, and then the reef was able to recover. That also could be, I mean, it's a bit different because it's been there for so long, but you also do see burnout at areas, like when you think about invasions or pathogens, you see that burnout at the epicenter, where they just reach max capacity and they're either resource limited or perhaps have density dependent mortality that occurs. It's sort of difficult to capture that with what we have, but that's also another possibility. Travis?
Can you talk about timescales of recovering from global stressors and local stressors and the difference then with the, like, talking about the human sort of impact there, where you went from sort of a microbial level to like the ecosystem?
Yeah.
And there's so many timescales.
Yeah. So for this local stressor, with such a large scale shift, I think you're looking at a longer time scale. That being said, there are places like in Hawaii and some of the bays there where you have those big blooms of invasive algae that come in and bloom and then go away. This sort of shift that has had such persistence throughout time, I think you're looking at a longer time scale for recovery. I was amazed. I mean, when I thought it would be cool to set up those transects in 2013 to complement Jen's original one at that site, I did not think that I would capture such a strong, I mean, going from, 70% groundwater cover on average to like 3% cover on average over 2013 to 17 is crazy. And yeah, I think that in general is going to be longer. And then for recovery, thinking about global stressors, I think it really depends on the mortality that you see. Palmyra, I didn't go into this a lot just for the sake of time, but Palmyra had widespread bleaching but really low mortality. And on the reef terrace, we actually saw almost no decrease in hard coral cover as a result of the bleaching event. On the coral reef, there's about a 9 or 10% decrease. And we think that might have to do with the natural variability in temperature on the reef terrace on a regular basis. It's a really dynamic environment, and so those corals might be better adapted to changing temperatures and temperature fluctuation, which is part of what we were looking for in that microbial community as well. But I think in areas like those parts of the Great Barrier Reef or Jarvis Island also suffered massive mortality during this last bleaching event. But they've already started seeing, Jem and I were just in on a conversation where Jarvis had a lot of Montifera. It all died. There was a huge, increase in turf algae, but when they were just out there for the NOAA crews looking at the community, it's actually mostly CCA now, and there's baby coral settling on that CCA. So I think you do have that possibility for relatively rapid recovery from stressors in ecosystems that don't have all of those other impacts. Abby?
It seems like in invasion ecology, the native species are sometimes in the unfortunate situation where they're facilitating the invaders in some way. You did mention that the coral morph does better, or it likes to settle on algae or CCA.
Yeah.
Did you ever notice it was somehow more successful growing next to coral than growing surrounded by a bunch of other coral morphs?
No, but the way the coral morph grows, it tends to start as that one little patch, and then that spread from it is so fast that typically if a coral morph is growing next to another coral morph, they're literally at such high density in those areas that they almost like overlap each other, like they push up against one another, and it's just we call it the shag curvet. And the coral morph growing next to corals, if there were just a few polyps, they don't do quite as well. They seem to do really well on sites like some of those pictures I showed where there's a lot more turf and then the pink CCA, the high wave action or whatever, the coral morph does really well at those sites where it can just kind of rapidly grow over CCA and the turf algae. But it is, I mean, as they grow over the coral, we don't really know. They could be, using these material filaments and digesting some of the coral tissue and getting energy from them that way. We don't really know that for sure.
So I have to sort of first say that I'm more of a microbial ignoramus. Peter had to explain to me what those axes were in the microbial plots. But sort of an off-the-wall question is, when people are having problems with their biome.
Mhm.
They often take probiotics.
Yeah.
So could we administer probiotics to the reef and help it recuperate
Yeah.
faster?
People have explored that option.
Right.
And actually, I often draw that analogy to like our gut microbiome when I'm talking about the coral microbiome. People have tried that. People also put antibiotics on and looked at the shifts that occur when you expose the corals to antibiotics and how they're able to handle stress. I think the probiotic idea is cool, and there's been work thinking about it kind of inoculating corals with good bacteria or good symbionts that are more thermally resistant.
Yeah.
And Ruth Gates, who recently passed, did a lot of work looking at thermally resistant corals and thinking about seeding them out on reefs to provide a more tolerant ecosystem. I think those are all really cool ideas. I think it's important that those ideas get explored. I also think that the large-scale feasibility of saving a lot of coral and a lot of reefs doing that is minimal, if at all possible. The idea of having the banks, which actually the Navy has been exploring a little bit too, having banks of coral species and bringing up ones that are more thermally tolerant and having those as this kind of backup for if we mess things up too badly might be one way of thinking about it. But if you think about the size of a coral reef and you think about trying to inoculate it with probiotics, it's pretty, I can't imagine that working that well.
And you gave a great presentation.
Thank you. Thank you very much.
Any other questions? Kasha? I was curious about some of the image analysis that you and your lab
Yeah.
do. You guys must come back with so many photos. And I'm curious if you analyze that manually or if there's some automation.
It's funny that you should ask that. It depends which coral, or it depends on which image you're talking about. There are two different things that I talked about in here. One is the photo analysis from those photo quadrats. This is actually what I started doing for Jen 10 years ago from all the photos taken from the NOAA Line Island, or from the NOAA cruises all around the world, where you take that photo of the photo quadrat, you upload it into this software, you put 100 stratified random points across it, and then I manually go through and identify everything underneath each one of those points. And so for the photo quadrat data that I presented here, that was primarily me that did that, as well as some other volunteers and other lab members in the lab, and Jen herself as well. So that is very, it is manual, it is pretty labor intensive. However, this just requires having the knowledge of benthic organisms so that you can accurately identify stuff. What takes longer is creating those photo mosaics. And that is a huge undertaking that a lot of people in our lab are a part of, and 100 Island Challenge does a lot of that work as well, because it's thousands and thousands of images that are brought back and then put into specialized software that stitches them all together and finds those common points. And a lot of those are actually made into 3D models as well. Although I didn't work on 3D models at all. But that was cool. It let us look at changes in the, I didn't have time to present all the mosaic data, but we were able to look at changes in the patch dynamics of the corral morph, both as they broke up and also as they expanded at 2 of those sites. Oh, yeah.
Great presentation.
Thank you.
So my question with the microbiome data.
Yeah.
Do you have a sense if you show those great shifts in the communities from the stress environments, if there's a when a coral generally is stressed, does the microbes change, have a higher similarity to like the seawater or the sediment, or do you think it could be an enrichment of their existing host-associated microbes?
Yeah.
Could it be to like other corals that are proximal to it?
Yeah, I think that people have found sort of a number of those different things. There's a little bit of the chicken or the egg with some of the stress microbiome. Like, do some of the bacterial families that live there die out, and then opportunistic bacteria from the water column can come in and settle and take advantage of that space? That is seen pretty regularly. Also, though, corals do naturally have some of those pathogenic bacteria present in low abundance on their healthy tissue. Like, you see vibrios on corals that are perfectly healthy and in really, unimpacted ecosystems. And so the other idea is that that stressor kills off some of the the good bacteria on that coral microbial surface, and then some of those vibrios maybe could expand, or some of the other pathogenic bacteria as well. I took water samples in conjunction with these, but those didn't end up getting processed just because of processing and funding limitations. So I'm not really sure in this case if the introduction of some of the rhodio or flavonoid came from the water column, or if they just were present in lower abundance in the first place. But each one of these, I mean, the data from that data set is like overwhelming, right? I mean, there's so many bacterial families, and especially the Montipora have hundreds of them present on them in all different states, and just like the abundance of them changes depending on the stressors and throughout time.
Okay, well, let's thank Amanda. Thank all of you again for joining us today. We'd like to invite you all to join us at Surfside in probably about an hour if you are interested. Otherwise, I'd like to keep the committee back behind so we can further question that.
Yeah, fine with me. Works for me.